Good evening, everyone. This is our seventh Los Alamos Faith and Science, or eighth, I've lost count, meeting. Uh, so basically, we'd like to thank John Abraziano and Robert and Susan Deraja for dinner tonight. And so anyway, uh, there's a kind of an interesting story about when John was coming up here. He wrote an email at one time. His plane arrived like about 10 o'clock, so he started driving up here at 10 o'clock last night. And he says, "Is it safe to drive up here at that time?" And I says, "Yes, we have guardrails." And so, he drove up at night, so he had no idea he was going by. So I talked to the hotel, and I said, "Okay, so when can you put him on the? He was at the Holiday Inn Express, So I basically said, so when you place him in a room, make sure you put him on the third floor and facing the canyon, where the floor is 300 feet below. So when he throws open the curtains in the morning, he'll have a wonderful surprise. Anyway, all right. So John is going to be talking to us this evening about faith, science, biotechnologies, and navigating science and integrity. John Loike is an internationally recognized scientist, educator, and bioethicist whose career spans decades of groundbreaking work at the intersection of biomedical research and ethics. He holds a PhD from the Einstein College of Medicine of Yeshiva University and completed postdoctoral research at Rockefeller University and Columbia University College of Physicians and Surgeons. Currently, Dr. Loike serves as professor of biology and bioethics at Touro University and co-director of the Master's of Science degree and graduate certificate program in medical ethics and humanities at New York Medical College. He is a prolific author of peer-reviewed publications in leading international journals addressing frontier issues such as stem cell research, cloning, CRISPR, gene editing, and the moral boundaries of biotechnology. Dr. Loike's research focuses on the ethical dimensions of emerging medical technologies and has shaped global conversations on bioethics and biomedical policy. He is frequently invited as a keynote speaker and inviting scholar at prestigious universities and institutions around the world, contributing deep insight into the ethical framework and the governing scientific innovations. His recent book, Science-Based Ethics, embodies his unique approach to bridging religious, philosophical, and scientific perspectives on vital topics including organ transplantation, reproductive technologies, and embryonic cell research. Dr. Loike's work continues to inspire ethical reflection and dialogue in both academic and clinical settings. So let's thank John Loike for coming.
It's amazing what AI can write about you that you don't know. Thank you. First off, I have to thank all of you. This is really a real honor for me to be here. It's one of these conferences where I got to see more than just come and present. I got to see your beautiful country, meet some of you. It's so interesting, your jobs and your careers. It's really louder. Can you hear me now? Can we make it louder?
Louder?
Yeah.
Okay.
All right. Oh, that's good. All right. Anyways, and I want to especially give thanks to Kirk, who's been an unbelievable host. Really taking me around, showing stuff. It's a real pleasure. So thank you very much. I really appreciate it. And I'm having a great time. I'm not ready to leave yet. Okay. If I say a term or I say something that you don't understand, don't hesitate to interrupt me. You can ask me questions. Hopefully I'll be able to answer some of them. And we're going to do something which Kirk told me about, so I think it's going to be interesting. After my talk, I'm going to give you a case. And around the table, you're going to review that case, come up with your own creative ideas how to resolve the bioethical dilemma, and then we can have a nice discussion to see your creativity, which you can't get on AI. Okay, so let's start. Yeah. There we go. So this is really what it comes out to. Are we playing God in balancing our ability to be innovative and our ability to have responsibility for what we do? And this is very, very challenging ethical dilemma. What's interesting about the idea of playing God on a secular bioethical terms is everybody uses it. Every secular bioethics, if you read a paper, they'll say, oh, if you use this technology, are you playing God? Even though they may not be religious and they may not even believe in a God, but it's a very catchy phrase. So what I'm going to do today, I'm going to just focus on two biotechnologies, tell you a little bit about their background, the power that they can elicit, and some of the challenging bioethical dilemmas that emerge from these technologies. So the ones I'm going to focus on are stem cell biotechnology and the molecular biology of our genes, specifically gene editing. We are in a stage now that I can basically rewrite your genetic code. It's an amazing power that we have. And it has a lot of benefits. And as you will see, there's a lot of controversies associated with that. So this is the focus of the talk. How can we use these technologies ethically to improve the world rather than too harmful? And I will try to give you some of the Jewish perspectives on this, because this has been a topic in Jewish law and philosophy for thousands of years, which is very interesting for me. Okay, now the first principle is a very important principle in bioethics. Just because we can do something, we have the capacity, doesn't mean we should. And this is a very, very powerful message, but very difficult to define. And I'll give you some examples and you can react to that. So how do we know about stem cells? We've known, what is a stem cell? I'll tell you that in a few minutes. A stem cell is a cell, in your body, every organ has a cell that is designed to replace any diseased or any destroyed cells, liver, lung, brain, bone marrow. And we knew about stem cells for almost over 100 years. Why? Because everybody knew if you cut yourself, your skin has a cut. And the fibroblast skin cells, they grow back, they regenerate new skin cells, and you're fine. We know this in terms of your bone marrow, right? If you lose blood, you can regenerate the blood. And this is all mediated by these cells in your body that have the capacity to replace cells. Now, that's adult stem cells. And there are also what we call embryonic stem cells. I'll talk to you about that in a few minutes. So we knew about stem cells for many, many, many decades. And the properties there are two properties of a stem cell. Stem cells have the ability to divide and renew themselves as stem cells. So in your bone marrow, you have a stem cell that can continue to replicate and make more stem cells. Or the stem cell with appropriate chemical signaling can divide into two daughter cells, one of which is a stem cell, and the other is a cell that can differentiate. So in the bone marrow, you need more red blood cells, so the stem cell will now divide, create a new stem cell, and create a cell which will make more red blood cells. Or if you need more monocytes, or if you need more neurons in your brain, or more liver, each of the organs have this capacity with the stem cells. It's a very, very interesting thing, and that's shown in the slide. What's now very recent is we have, under certain conditions, you can take a stem cell and can make two different types of specialized cells. This is innovative, novel, we don't understand the mechanism, but could be very, very important in the future in terms of medicine, as you will see in a few minutes. So this is on a molecular level or a cellular level, you have what's called asymmetrical division and symmetrical division. Asymmetrical division, the stem cell forms into a stem cell and a differentiated cell. Symmetrical, it forms into two stem cells or two differentiated cells. Now, this is important because if we understand the signaling mechanism of these two processes, it will be a tremendous benefit in medicines in ways which I will tell in a few minutes. Okay, now there are three types of stem cells. This is a little basic biology. One is called a totipotent stem cell. A totipotent stem cell is, of course, the fertilized egg. And the first 8 divisions, until it gets to be a morula, all those cells from those first 8 divisions, those 8-cell pre-embryo, I can dissect them off and create a whole human being from those stem cells. Amazing. The second type is called the pluripotent stem cell. This is a cell that is only designed to make all the specialized cells of your body. A totipotent stem cell appears in the blastula, which is about 8 or 9 days after fertilization. It's in a specific area of the blastula called the inner cell mass, and I can take each one of those stem cells and I can make nerve, muscle, eyes, heart, you name it, but I cannot make an embryo. It does not make the cells to make a placenta and does not make the cells to make an embryo. So it's a very important type of stem cell, very powerful, and the basis of many, many of the therapies that I will describe to you, but it's limited. And the third type is called a pluripotent stem cell, or multipotent stem cell. This is the cell of the organ. The liver has its own unique stem cell that makes liver cells. The pancreas has its own unique type of cells, and they make all the various types of the cells that compose the pancreas. And what's amazing about all this is a summary of what it is. And here you see, where's the pointer? Let's see. Where's the pointer? Oh, doesn't work. Anyways, the totipotent, that's a fertilized egg. Here the pluripotent cell, this is the blastula. So that inner cell mass, it's about 128 cells. The cells surrounding that embryonic stem cells, those are the cells called the trophoblast. They make the placenta and the umbilical cord for the fetus. And what's amazing, it looks like just a cell mass, but the location of each one of those cells determines what type of cell it's going to come into. The one all the way on the right may turn into a muscle cell. The one all the way on the left it's all location, location, location. Stem cells function and they differentiate based on not only the chemical signals, but where they are located. And of course, the multipotent stem cell comes from the blood, all big red blood cells. Every organ has its own thing. Okay, how do you regulate this? So on a molecular level, what are the signals? So we have 2 different types of signals. One is called transcription factors. These are amazing proteins. We have about 1,600 transcription factors, and they tell the cell which gene to turn on and which genes to turn off. So we have 25,000 genes that encode for proteins. And all your genes of the cells, all the cells have the same genes. So why is a muscle has some cells turned on certain genes, and a nerve cell has others? That's all regulated by these transcription factors. These proteins say, you're designed to be a muscle cell. I will turn on about 9,000 sets of these genes, and to another stem cell will say, oh, you want to be a nerve cell, we're going to turn on a different set of about 10,000 genes. So even though the DNA in all of your cells are the same, it's these transcription factors which tell the cell its function, its destiny, and what it's going to be. They're amazing. And the more we learn about these transcription factors, the more we're going to have an understanding of what makes us who we are and what makes us unique. The other biological are called microRNAs. When I was in high school, we learned the DNA, your genome has about 3 billion letters. Very nice. But the 25,000 genes only composes 2% of the whole genome. What's the rest of it? So in high school, I was taught that's called junk DNA. But understand something, evolution does not produce junk. We only can say, God doesn't produce junk, evolution, depending on your religious perspective. And we're now learning that whole 98% of the genome, that we didn't know what it does, produce these small RNAs, which again regulate how the stem cells are going to differentiate and how they're going to function. And this is a whole new area over the last few years. And the combination of understanding transcription factors and micro RNAs will enable us to take any of your stem cells, and I can make that into any cell I want. Any cell I want, which is really very powerful. The other important thing to recognize is that we have building block genes in the body. They're called Hox genes. Hox genes tell you have to have 2 eyes, 1 nose, 2 arms, 2 legs. They come in. They're all located generally on 1 chromosome, and they provide the blueprint of how your body's going to be formed. And what's amazing about this is that we can do incredible things, not in animals, more in insects. I can take a fruit fly and have it grow its legs as antennas, or as antennas as legs, by modulating these Hox genes. We can do amazing things by regulating where these genes are and how they function. So this is how your body is composed of. Why you look like, what your cells do, it's all regulated by these proteins and these genes. They're very similar to all animals, and I'll tell you in a few minutes how we can really use them in very innovative ways. Okay, so let me again just briefly take you through this, that you have the fertilized egg, which is the gold standard of a stem cell. You have what's called the totipotent cell. Each one of those cells can form an embryo, and this is very important for a very important reason. We have genetic diseases. A woman is pregnant, right? Let's say, forget it. If a woman is a carrier for cystic fibrosis and her partner's a carrier of cystic fibrosis, she has a 25% chance of having children who are affected by the disease. We have now in vitro fertilization. We take this woman's oocytes, and we fertilize the egg with the partner's sperm. We generate in 4 days these totipotent cells, and we can remove 1 out of these 8 cells. It doesn't affect the biology. Those 7 cells are sufficient to grow a healthy embryo, and we take that 1 cell, take it to the laboratory, do genetics to say, is that embryo carrying both genes for the disease trait, 1 gene a carrier, or no genes? So we have a way to screen, almost 99% effective, for all of these fertilized eggs to determine which one you want to implant. And this has been an amazing breakthrough in controlling diseases like Tay-Sachs disease. Well, I mean, we rarely see Tay-Sachs. Cystic fibrosis, we rarely see it. We do see it, but it's expensive. But if this were offered, we could cut down so many. We have over 5,000 genetic diseases that we can apply this technique. And then, of course, from the totipotent cell that forms to the blastula, shown there, that's the inner cell mass of the green. And those are the cells that scientists like to use to look at stem cell therapy. They like to take these inner cell mass, these pluripotent cells, and work with them in the laboratory to do it. And then, of course, you have the blastocyst, which is shown here. And this is what you implant in the uterus. Now, this is very, very important. The embryo is a foreign body. The woman carrying the embryo should immunologically reject that fetus. It's foreign. It has genes from one partner, genes from the other. How does it survive? And over the last few years, we've discovered several, you were probably taught, well, a woman who's pregnant, she has different hormones, they're a little bit immunosuppressive, they tolerate more, instructions of foreignness. That's true. But what's amazing about the blastocyst is that there's a cell we just discovered in the uterus. It's called a natural killer cell. We knew about this for decades. A natural killer cell is in your blood. It's one of the best cells to stop you from having cancer. It's an amazing surveyor of cancer cells. It can identify, it doesn't have to be educated, it doesn't have to go to college, university. It can identify the cancer cell and it can attack it. But we realized that the uterus has these natural killer cells. What is the uterus doing with the natural killer cells? And we discovered, I say royally, scientists, not me personally, we discovered that these natural killer cells reformat the uterus of the woman to allow the uterus to immunologically accept the foreign body. That means that if a woman is pregnant, I can take her husband or partner's skin, transplant it onto her arm, and she won't reject it. She'll accept her partner's skin. It's a tough way of doing paternity tests, a lot easier way, but it does work. But it's amazing that it can do that. And the other thing is that these stem cells are highly mobile. So what it means is a woman is pregnant with a fetus, these stem cells leave the fetus, they cross the placenta, and they embed in the woman for decades, if not a lifetime. I can take a woman who's had a male child, and if you take a biopsy on certain muscle, parts of her brain, I see XY chromosomes. How do you have XY chromosomes? She's a woman, she should have all XX. But no, these stem cells come in, they're tolerated, they help the woman tolerate this foreign body, and they're there for life. And what their effect is on the behavior, on the disease of the woman, is an unknown area, uncharted. We know the opposite, that stem cells from the pregnant woman cross the placenta and end up in the fetus, in the baby. And how do we know this? Because sometimes a baby is born with a tumor. It happens. And they excise the tumor and they do genetic analysis. And guess what? That tumor did not come from the baby. It came from the mother, from her stem cells. So these stem cells are very mobile and they're very important in embryology. Okay. So this is a small video.
Embryonic stem cells, the most pluripotent of all stem cells, are derived from embryos generated by in vitro fertilization. When fertilization is successful, the sperm head bearing the nucleus enters the egg, while the tail is left behind. The egg divides first into 2 cells, then into 4. With more divisions, a multicellular ball of cells known as a blastocyst is formed. If you could look into the blastocyst with X-ray eyes, you'd discover that it is a hollow ball made up of 2 cell types, an outer layer, the trophoblast, which will eventually form the placenta, and an inner cluster of cells known as the inner cell mass. The embryo is derived from the cells of the inner cell mass. It is possible to pick up these embryonic stem cells with a pipette and transfer them to a petri dish for culturing. Under appropriate culture conditions, these embryonic stem cells divide or self-renew, and the cell mass grows. Sometimes pockets of cells will stop dividing and begin to differentiate spontaneously. Groups of cells may develop properties of mature bone cells or of pancreatic cells. Others develop into muscle cells that can contract, and still others into nerve cells. Because they have the potential to become a wide variety of specialized cells, embryonic stem cells are described as pluripotent. Pluripotency is one of 2 key features of embryonic stem cells. The second key feature of embryonic stem cells is their ability to divide or self-renew indefinitely while retaining their undifferentiated pluripotent state. Small groups of cells can first be grown in the petri dish. But as the numbers increase, the population can be further expanded by growing in larger tissue culture flasks. You can tell that the cells are growing because the tissue culture medium turns from pink to yellow-orange, indicating a drop in pH. Cells from a single flask can seed many flasks. In this way, unlimited numbers of undifferentiated pluripotent stem cells can be produced.
That's a quick summary. Now, the importance of this is in part recognized by the Nobel Prize going to John Gurdon, who really did a lot in stem cell technology, and Dr. Yamanaka. Brilliant. He was able to do something amazing. I told you about these transcription factors. He realized by just brute force, he could add 3 or 4 transcription factors and convert an adult cell into an embryonic stem cell. So I can convert your skin cell into an embryonic stem cell, make it into a liver cell, make it into a muscle cell, whatever I want. And by, again, manipulating these microRNAs and these transcription factors, I can make any cell I want. It's an amazing technology. So let's see, how does that, we'll skip this. I'll skip this, all right? Because we have a lot to do. So what is it used for? Of course it's used for experimental model systems, including we can do drug testing in terms of how it works on muscle cells we grow in the lab or heart cells, et cetera. But now the real big thing is what's called cell-based therapy. And here's an example. We all know what diabetes is. Much of diabetes is, of course, a genetic, part of it is an immunological deficit. What happens in diabetes? The pancreas has a cell called the beta cell. The beta cell is destroyed by the patient's immune system, by specific cells called killer T cells. And now, without the beta cells functioning, it can't measure your glucose level, it can't make insulin. Insulin is a very important hormone, as you all know. It allows the body to maintain homeostasis of glucose, which is critical in any life. People with type 1 diabetes, they have a hard time. They have to take insulin shots, they have a lot of diseases, it's very difficult. With stem cell technology, we have an amazing cure that's just over the last year, which is going to get FDA approved if it hasn't already, which is amazing. And this is as follows. You would say, well, you've destroyed the beta cell. Let's take another cell from the pancreas that we'll call the exocrine cell, convert that to a new beta cell, and inject new beta cells into the patient. You could do that, but the problem is you haven't fixed the immune system. So the immune system is going to destroy those newly implanted beta cells, and you're back to the diabetes one. So the group at Harvard did an amazing thing. What they did is they took the stem cells that they now converted into fresh, new beta cells from the same patient. You don't have to do any it's all autologous transplantation, and they encapsulated it into a nanoparticle. The encapsulation allows glucose to come in, so they can sensor glucose levels, and allows insulin to be released. But it's encapsulated. So the killer T cell that normally has to attach and kill the beta cell does not gain access. So they implanted these particles, nanoparticles, and they can cure the patients from type 1 diabetes. Isn't that amazing? It's expensive, but it works. It's an amazing, amazing technology. So that's an example of cell therapy using stem cells. All right, cloning. We can take stem cells and clone. We've cloned dogs, cats, and anybody if they've ever lost a pet, $50,000, that's all you need. And we can clone your cat. What's really interesting about that, speaking to people who've done that, the personality of the cloned dog or cat does not match the original, which is fascinating. But it's doable. It's really doable. And we call cloning somatic cell nuclear transfer because it doesn't sound as ethically challenging. It sounds more neutral. But basically, the way you do it is like how they did for Dolly the sheep. Is what they did is they harvested oocytes from one type of a sheep, and then they removed the nucleus, which contains most of the DNA, from that oocyte. And then they took a somatic cell from an animal to be cloned. They took that nucleus of the cloned animal and transplanted it into the oocytes. They gave it a little electrical shock, set it going, and guess what? They cloned a whole dolly. It was amazing. The paper in 1997, Cloning a Dolly, was the most fascinating paper I ever read in my life, in terms of my lifetime, I should say. I was mesmerized by it. And I happen to have had the privilege, when I saw the paper, I read it many times, I realized they didn't look, DNA is not only found in the nucleus, it's found in the mitochondria. So I said they didn't look at the mitochondria. So I wrote to Ian Wilmot, who was a senior author, and I said, send me some of Dolly and we'll do it. And we published a paper on the origins of the mitochondrial DNA. Amazing, amazing. And because of that, I began writing about ethics, and I was invited. There was a huge conference in Edinburgh It was like the Oscars. Everyone came in tux to talk about. Gordon was there, Yamanaka, everyone was there. And it was an amazing thing to watch, everything. So, and actually, I had read all the papers, I talked to them on the phone, but meeting them in person was a whole different genre, very, very exciting. Okay, applications of cloning, let's go through. So, pets I told you about. We can clone human beings, absolutely easy. Expensive. Now, of course, if you ask me, who would I clone? And I would say, LeBron James, right? Clone LeBron James, make $10 million investment, and in 20 years, I have a basketball franchise worth $5 billion, right? We can clone human embryos with stem cells rather than sperm and egg, and that's going to be the case you're going to discuss at the table. What is the ethics of that? No need for any more sperm. No need for any oocytes, eggs. I can take your skin, your blood. I can take the hair off your head and isolate the stem cell of the hair follicle and make it to a gamete. Take hair of somebody else in China or in Nepal, put them together. All I need is a gestational surrogate. We have a human being. Pretty powerful. And we can create organoids. This is amazing. We can create synthetic ovaries, synthetic livers, lungs, and the ovary is interesting. Listen to this. You have a woman, a young girl, 9-year-old, this is really true, 9-year-old girl, she comes down with a leukemia. A 9-year-old, if you want to cure her, put her on intensive chemotherapy. That's classical, and within 4 years, she's pretty much cured. But the chemotherapy, what is it going to do? It's going to destroy all her oocytes. She will be infertile. Before this, what did they used to do? They used to take out the ovary from the young girl, freeze the tissue, hope that none of the cancer cells were there, and reimplant it. It's challenging, but works. Not today. Today, what we can do is as follows. We take her ovary out. We remove all the cells from her ovary. We're left with this architectural scaffolding, biological scaffold. We cure her of the leukemia, no leukemia. We take her blood cells, convert them to ovary-like cells, implant them on the scaffolding of an ovary, and because of location in the matrix, it forms a complete ovary, producing oocytes. We implant it into the arm of her, into the under skin of her arm. Why? Because it's easy to do ultrasound. She's going to ovulate every 2 months, and when she's ready to have children, we remove the oocyte, do in vitro fertilization, and she can have healthy children. No chance of cancer. Isn't that amazing? This is the power of stem cells and organoids. Now, of course, you're thinking, wow, if that's true, what about the brain? We can make neural organoids. Very interesting. I was on a National Academy of Science think tank about the ethics behind that. Very difficult. Because on one hand, we know very little about our brain. On the other hand, this would be a tremendous tool, but what does it mean to create a neural organoid? I mean, think of what the implications of that. All right. We can make lab-grown human embryos, right? And they're doing that. Today, there's what's called the 14-day rule. If I make an embryo with sperm and egg, I cannot leave it in the lab for more than 14 days because of the ethical. When does it become personhood, humanhood? However, if I make it out of a stem cell, it doesn't follow the 14-day rule. I can get to look at embryos which are older than 14 days. That's very important. We know so much about mouse embryology. We know so little about human embryology because we don't have any good models. And miscarriages, we don't have a good understanding what causes a miscarriage. Creating these human embryos would be a tremendous answer. Is it ethical? Does the ends justify the means? And that's the organoids. And I told you about the artificial oocytes, which are amazing, amazing things. Okay, I want to introduce you to what's called the yuck factor. This is one of the most interesting, non-academic definitions I've ever come across. It's by Leon Kass, brilliant guy. And he says it's the idea that our instinctive feelings of disgust or repulsion can serve as a moral guide. And his examples, he says, listen, we go to the Met, you go to the Louvre, you go to the Uffizi, you see a lot of nude. Is that pornography? How do you differentiate between pornography and legitimate art? There's no definition. It's all what you feel. It's the same with the gut. The yuck factor is that you look at this and say, do I want to clone a human being? That's yucky. There's something, I can't tell you why, but it's a yuck factor. And that plays a role in bioethics, believe it or not, even though there's no good definition. Okay, so here's the case that you're going to think about. Rather than creating a human being with sperm and eggs, we're going to do in vitro gametogenesis. We are going to create an embryo from skin and from blood. What are the ethical ramifications? What are the challenges? That's what you're going to think about after this talk, and you can give me your thoughts. Interesting topic? All right, good. Okay, so once the sperm, they could begin use IVF and ingestion. Okay. Now, what is the religious scholars' views of synthetic embryos? Let's give you a little perspective. So, Catholic scholars believe that the human reproduction requires marital relation. They are against gestational surrogacy. They're against in vitro fertilization because they feel human production requires marital relations. And if you don't have that marital relationship, it's not within the parameters of a religious human being. Very interesting. The Jewish view is a little bit different. The Jewish view says creating life through natural means, even without human reproduction, is not prohibited. The importance here is getting that human life. How you get to it can be religiously allowed. Why? Because there's a principle that we as human beings, God, in the Jewish perspective, the world was created incompletely. We are partners in the creation process, and we have to partner with God to improve and perfect the world. That's called Tikkun Olam. And it's not universal, it's not like it's global, but we have all these guidelines of how we ensure that what we do is part of this partnership with the divine to improve the world. Okay? Now, what's interesting is that the legends, the Jewish legends, says that when God first created a human being, he took him and led him around all the trees of the Garden of Eden and said to him, see my works, how beautiful and praiseworthy they are. And everything I've created, I made for you. Be careful not to corrupt my world, for if you do, there will be no one to repair it. So that is the limitation of Tikkun Olam. You can be partners, but make sure you don't destroy the world with the technology that you are examining. Okay, that's the first half of the talk. The second half, I'll have to go a little faster. We're going to now go on to gene therapy. Gene therapy is our capacity to rewrite your genetic code. It is amazing. I will show you, it has so much applications in agriculture, drug discovery, industrial biochemistry, gene drives I'll tell you about, human therapeutics. It's called CRISPR, and it's an amazing technology. It is cheapy cheap, very fast, and the only limitation now is you want to make sure that you're editing the right part of the genome. Off-site editing is a little bit of a problem, and even though we're at 99.99% accurate, when you think of 3 billion bases, if you have 10 or 20 or 30 mutations, that can be terrible. So it's not there yet, but it's getting there at an amazing pace. And there are different things you can do with it. Sherlock, it's just amazing. Sherlock is using this gene editing capacity. I can screen all of you for multiple infectious diseases. I can screen you for COVID, for measles, for smallpox. One blood sample on a thing, and I can screen you cheapy, $10 a screen, and it's very efficient. Then there's the Pac-Man idea. We can take certain stem cells, certain genes, and using CRISPR, we can make them into aggressive, killing machines that can destroy your cancer like a Pac-Man, coming, eating it up, all the cancer cells. Specific for the cancer cells, doesn't affect healthy cells. We have, and of course, one of the most controversial things, we can use gene technology to create synthetic nucleosides. I can make a synthetic, unnatural genome. Why would I do that? Well, I would do that because A, I would like to have a better bioinformatic system to make biocomputers. The more elements I have in that, the better it's going to be. What scares me about creating synthetic nucleotides that they've done it, put it into bacteria, the bacteria replicate, they make new types of proteins, new types of genes. What's scary about that is that in your cells, you have 160 proteins whose sole function is to make sure your DNA doesn't make any mistakes. 160. These are, if you had time, I'd show you videos how they survey the cell, how they attach to the right part, how they excise the mutated gene parts, how they repair it. It's amazing. And what's amazing is that if it finds a mistake, it gives the cell 24 hours to fix it. If the cell cannot fix the mistake, the genetic mistake, it triggers what I call cellular euthanasia, the apoptosis, the cell kills itself. Now, these 160 proteins are amazing, but they don't recognize synthetic nucleotides. And that scares me. Without the capacity to repair these things, you can begin to think of bioweapons and diseases that would just are frightening to think about. So that's my concern about that. CRISPR was discovered by Chevalier and Jennifer Doudna. It's a very interesting history, and they really did not know what they got their hands into when they first discovered CRISPR, but pretty sure they figured it out.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, what are the ethical concerns? First of all, one of the things is that, are we playing God? Are we really changing an evolutionary, well-tested genome for billions of years, and we're changing it? And not only can we change it for medical purposes, but we can use it for other ways as well. For example, we can do what's called gene drive, that I can take a female mosquito and disable that mosquito from ever replicating. Female mosquitoes that carry malaria reproduce once with the male and will never reproduce, will never mate again, and lays all the eggs and causes the malaria. I can take gene drive and disable the fertility of that mosquito so that every generation gets that same mutation. So I can eliminate a whole species of malaria, of mosquitoes that carry malaria. Should we be doing that? We are. In the islands in the Pacific and the Caribbean, we are eliminating these mosquitoes to see what are the ecological effects. This is really, if you think climate control is challenging, ecological regulation is even more challenging. So I don't know, but we can do that. We can also.
Thanks for brilliant for supporting.
We can also create woolly mammoths. They're doing this. They've taken the genome of a woolly mammoth. We have from frozen, we have the DNA, it's all intact, and we know the differences between Asian elephants and woolly mammoth elephants. And they've now constructed an Asian elephant genome and converted that to woolly mammoth. We can now, in the next few years, use that to create an embryo with a woolly mammoth genome, put it in an Asian elephant, and give birth to woolly mammoth. Why would we do this? Because one says, well, A, you want to know what it's like. It's a good thing to do. The other thing is that woolly mammoths were very good in ecology. And they are in the tundra and Russia, the way they eat the vegetation, they leave some residue. So it's very good for purifying the air. And it could be a very effective way for controlling climate. But so they can do that using stem cells and CRISPR. Also, we can do that with human beings as well. We have the DNA of the Neanderthal. We know. And we can take, I don't know if you're interested, we can take a woman and say, we'll implant you with a genome of a Neanderthal, and you'll be the first woman to give birth to a Neanderthal person. Is that ethical? Why would we do it scientifically? Well, because, what were they susceptible to, what diseases, what were their strengths, what were their cognitive functions like? A lot of things we could learn. When I presented this to my class at Columbia, they said, can't do one. You have to have a community. Who are they going to marry? Who are they going to date? Well, that's how Columbia students think. But we can do that. Is that ethical? And one has to think very carefully. If we as human beings destroy the species, because hunters, rhinos, right, or elephants, then maybe we have a moral obligation to revive. But if a species naturally was destroyed, maybe there's a reason for that. An embryological, evolutionary reason, and maybe we don't have the right to reinstate that species. So these are really ethical debates. Okay, so we can make dogs with unbelievable muscles, and we're using that, right, to apply that to diseases like muscular dystrophy. We can make cattle and bulls with so much mass, it'll really change our ability to the meat industry, and we can make babies. We can design babies any way we want. Now, medically, I'm sure most of you would say if there's a medical reason, we can do CRISPR. And I'll give you a concrete example that just came out last year. Sickle cell anemia. Do you all know what sickle cell anemia is? You have hemoglobin, one letter of that gene was changed, and now instead of the hemoglobin molecule being globular, it is fibril. And it causes sickle cell, the red blood cells to be sickle, and they have a shorter half-life, and you're always anemic. Very terrible disease. So a scientist about 20 years ago said, something's wrong here. If in sickle cell you have this anemia, how do the embryos survive pregnancy? Why aren't they destroyed? So he discovered that all embryos, they don't make adult hemoglobin. They make a special form of hemoglobin called fetal hemoglobin, which has a higher affinity for oxygen. That's how it can get oxygen from the mothers. And that is very effective. And once the baby is born, there's a gene that gets turned off to turn off the production of fetal hemoglobin, and another gene that gets turned on to make adult hemoglobin. So now they did something very clever. They used CRISPR and gene editing to disable that switch. So they could take somebody who's living 40 years with sickle cell anemia, and now they make that patient make fetal hemoglobin, and he's cured. I can show you video, it's amazing. The eyes are white, they're not full of bilirubin, they don't need transfusions, and it works. This is the power of medical. What about non-medical? Do you want your son, daughter, grandson, granddaughter to have an IQ of 200? There are 100 genes that regulate IQ. We could change all of them at once. It's not hard. We can make somebody have blonde hair, blue eyes, red hair. We could do so many things for non-medical reasons. Should we be doing this? So this is a real, real concern. So let's understand this, these unintended consequences. We can turn back the clock on aging. There are genes that, as you age, become active. There are, I don't want to go into details, polymerase genes and telomeres, and we can reverse that, and we can make all of you have a cellular fingerprint of a 20-year-old. Should we be doing that? We can target this, called the church genes, right? And we can make people, we're all unique. We have all unique genomes. Even identical twins do not have the same genome. Because as soon as the embryo splits, mutations happen, and the genomes are different. That's why if you know any identical twins, sometimes they have different color hair, different color eyes. They're different. Are we going to erode our human identity? Okay. And the other ethical concerns is the idea of equity and access. They're very, very expensive technologies. So the drug companies come up with something very clever. I'm very impressed. We call this the mortgage treatment of therapy. So using stem cells to cure sickle cell or to cure CRISPR with sickle cell or to cure type 1 diabetes, about $1 million to $2 million per person. That's a lot of money, right? So what they do is as follows. They'll say that the government will sponsor the first round, which hundreds of thousands of patients, and the drug companies and patients will have 20 or 30 years, like a mortgage, to pay it back. That's an effective way, possibly, to deal with the financial crunch of doing these therapies. Okay. So this is what it costs just to do some CRISPR technology. It really is incredibly expensive. All right, so what can we do? Increase human longevity. We can, of course, as I told you, use therapies. an alternative method, which is so interesting, is hyperbaric oxygen therapy. I don't know if you know this, but it turns out that from the space agency, from NASA, they made an amazing discovery. They had identical twins. They sent up one twin at a time. And when they came back, they did a whole biochemical analysis. And they realized that the twin that was up in the shuttle for 6 months, their telomeres, which are the ends of chromosome, and you should know that a telomere tells you how old you are. You're all born with telomeres, these end of chromosome sequences are 15,000, and every year they get shorter and shorter. When they reach 5,000, off to heaven you go, all right? But they realized that being up in a shuttle for 6 months, the telomeres increased in the twin up there versus the twin on Earth. And we know that if you put a person in a hyperbaric chamber, because what's the shuttle? Has to be hyperbaric to get the oxygen into the lungs. So in Thailand, they have 20 of these. They're doing clinical studies here in America. And for $10,000, you can buy in a hyperbaric chamber. An hour a day, they're easy. I've been in one. They're very easy. You come in there, an hour, you read a book, just, and that's it. Your telomere length will increase, and presumably you will live longer. I don't know, healthier, that's a different story, but you will live longer. Is it worth $10,000 to invest to live to 120? So now you don't have to retire for 60 years, you can work for 100 years and retire for another 60 years, You can do whatever you want. So what are the moral limits of genetic engineering? I mean, this cartoon says a lot. Some genetic engineers, some genetic engineer we turned out to be, right? I mean, we have a lot of power with this, and we have to know how to use it effectively. So, A, we have to respect autonomy and informed consent. If we're giving this to patients, they have to understand what this technology is. It's not easy. I mean, you have this problem. How do you explain physics to somebody like me who doesn't know anything about physics? It's a real problem. When you give a test, here a patient comes in, diagnosed with pancreatic cancer. He's going to live maybe a year or two, if not 3 months. And you can explain to him all about T cells and NK cells and all these different immunotherapies. And they're not going to understand it. They're going to turn to the physician and say, Doc, what would you do if you were my condition? I don't want to know about the science. I just want to know what to do. That's not real autonomy. Autonomy is you have to make a logical decision, you have to understand that. You can't give an exam that says, here's the literature, read it, come back, and if you pass the exam, we can tell you what to do. But it's really an important aspect of how to present it. How do you balance the potential benefits versus unforeseen harm in creating new forms of inequality? And how do we ensure that this access is not limited to privileged populations? And we see this happening all over America. The privileged have certain access that we don't, and that's not good. And then how do we define a normal and desirable gene trait? And how do we avoid the danger of eugenic thinking, the idea of creating what we want to do for non-medical reasons? These are some of the real ethical challenges. And then the principle of precaution. This is very important. We got to go slowly. We can't jump into this. If we go too fast, we don't know what we're going to end up with, because we're ending up with tremendous changes, dramatic changes in who we are. And can you regulate this? This is not a trivial question. They have do-it-yourself CRISPR that you can buy over the web. You can do it, it's easy. You can inject it, you can change whatever traits you want. How do you regulate all this stuff? How do you regulate so bioterrorists don't get it? These are real serious ethical things. And the question is there a moral compass in society? And I want to say something controversial. I think there is. I think there's a moral compass. And how do I know this? We've been doing in vitro fertilization for almost 50 years. Over 10 million babies have been born by taking sperm, egg, in vitro, selecting what we want, and putting it back. And there's not one case, one documented case of a designer baby. Not one. So maybe there is a moral compass in society. Not everybody uses it, but it might be genetically inherited in our genome. And the idea of moral limit that is extending longevity, a moral endeavor, or is it a violation of natural law? What does natural law mean? What does biology really mean in terms of making moral decisions? So Judaism idolizes scientific interventions on behalf of humankind, but there are limits. In Judaism, you can only violate, quote, unviolate natural law for the benefit, for medical benefits. You can't just do it willy-nilly. There's limited autonomy in what you can do. That's the philosophy. And human beings have to be careful when engaging in technology that could violate natural law. The world, whether you look at it from a religious perspective, evolutionary, the world has survived for so many billions of years or millions of years based on certain principles, some of which we understand, some of which we don't understand, and changing those, intervening with those, could have deleterious effects. And since one's death due to terminal cancer be reversed as an interventional action, I mean, this is a theological, medical question. There are a number of approaches to that. In Judaism, life is precious, and the question is that in Judaism, demands human beings engage in medical response to save a life. And this carries all the way across. So any technology that can save life is endorsed by the Judaic law. How do you resolve some of these ethical challenges? All right, how do you resolve consent and ownership? How do you resolve commercialization, exploitation? How do you establish international regulatory agencies? How do you incorporate AI-powered ethic review boards to automatically undergo these checks and balances? And can you use blockchain records as a way of privacy? Privacy is a huge issue. As you know from AI, we have no privacy anymore. There's no privacy. They can do, everybody knows, and once, from a medical perspective, I would say we should sequence every newborn embryo. And that should be made available. Because knowing your genetics is so important to knowing how you live a healthy life and how you deal with disease in the future. But who has the right to that information? How can we ensure that only you have that right and life insurance companies, health insurance companies, employment doesn't have those rights? How do you ensure privacy there? It's a really challenging thing. And should we have time banks, pause and review? Should we keep going? Very hard. Oh, you see from AI. It's very hard to stop technology. We can't. How do you regulate it? How do you stop it? These are really some really challenges. So what I've told today is that resolving ethical challenges is a very intelligently creative process that's not easy. And you can invoke the yuck factor if you want, out of the moral guides. And the idea of CRISPR and human hubris is really an idea of balancing what it means to be human and what it means to be not human. And again, equity and access and prevention. And this precautionary principle, the idea of balancing potential benefits against the risks, is something that there's a lot we don't know. There's a lot we don't know. How far do we go into this? And this is my book, if anybody wants it. It's on Amazon. It's a very unusual book, because as I wrote it for the Columbia students about 10 years ago, I had all of them make corrections. And every 2 years, I put out a new edition based on their input, which is fantastic. The problem is that when Oxford Press and Cambridge Press wanted to publish for me, I said, sure, but you got to revive it. You got to re-edit it every 2 years. I said, we can't do that. You can only put out a new edition every 5 years. So I didn't agree to that. So I self-publish it, but it's really based on the students' responses and their questions. It's an interesting book. All right, this is basically what I have to say in the hour. And this is the first case. You're going to hand out sheets? All right?
They're already on.
Oh, they are. So it's 7:30, right on time. Oh, so take, 15, 20 minutes? Talk among yourselves. I'll be around you. You can ask me questions. See what you can creatively generate to resolve this ethical issue. Should we be engaged? Yes?
I know that maybe this isn't the time to ask the question, but what exists now for regulation and control over what's going on in this kind of
Very little.
Okay, so there is no animal.
I mean, there are, I told the 14-day rule for embryos, there are some regulations, but it's very difficult. Are there any other questions here?
Hey, it's not. What's that active?
Excellent question. I don't know.
Oh, okay.
It's not a good ethical, it's not a good ethical result.
Okay. So next week, James Carroll will be presenting on ethics, love, and the alignment problem in artificial intelligence. So anyway.
How intimately
Oh, okay. Go ahead.
Excuse me, we can't hear the
Okay,
question.
the question is, how intimately involved are corporations and the profit motive in all of this? Greed and self-interest, they never go away.
No, it is involved. It is involved. It is.
They can still hear you.
Okay. Yes, it is a factor. Justifying the high cost of these therapies is a very challenging understanding. Remember, a drug company, you have to understand they fail. So when you have, I'll give you an example. When I first tried to make a drug to dilate the coronary arteries, Pfizer put in $100 million, and it failed miserably. They did for unbelievable stuff, and part of their structure is that, okay, we lost $100 million, we have enough. So this is one of their rationales why they can charge such exorbitant prices. What's interesting about this drug is that they hired a new postdoc, and he says, what do you do with all this data on these thousands, tens of thousands of patients? So we're going to just library. I said, why can't you look at it? Maybe we'll discover something. They said, all right, you have 6 months to look at the data. He looked at the data, he made an unbelievable observation. The patients taking this drug, called Viagra, had an enhanced erection for 4 to 6 hours. And he says, here you have a drug for erectile dysfunction. And Pfizer went back, they got FDA approval, and I need not tell you about Viagra. And what's interesting is not only that, they also discovered, this took many more years, don't ask why, not only does it enhance erectile dysfunction in men, but it stimulates sexual achievement in women as well. So women should be taking Viagra if they want to enhance their sexuality. So these are drugs that we would have just thrown out. But drug companies, part of their scheme to say, oh, we're not making money, we're making money, but we need the money for the failures. So that's their rationale. But yeah, there's profits. Any other questions?
Any other questions?
Yes.
Okay, go ahead.
Yeah, hi. Thanks for a very interesting talk. So you mentioned life extension, ways for people to live longer. And I'm wondering if, aside from any CRISPR technology or anything, if observational studies have been done of populations around the world where people live to 110 or 105,
Yes.
and how that might impact
Yes, it
the
does.
CRISPR. Yeah.
I mean, there are certain general parameters. I mean, we know about the Mediterranean diet. We know about exercise. We know about sleep. General things. There's no one magical cure, no one magical food that you can take. Although, if you take rapamycin, which is used in organ transplantation, people live longer because it's molecular biochemical effects. So that was a thing we didn't know when we first designed rapamycin, but it's so if you want to take with its side effects, you can live to 120, 150. I can't tell you how healthy you're going to be, but you can live longer.
So is that guiding some of the
It
current
is
thinking?
more on
what to do?
TikTok than anything else. But I mean, it's so interesting because I have a very keen interest in epigenetics, how lifestyle and food affect health. So I go into a lot of details about these studies. And some of the studies are really good. Most are not good at all. But there is a lot of evidence that it's all individual. It depends on your system. How you respond to a food is going to be different than I. Again, it goes back to the genetics. We are so different that you may benefit by eating blueberries, strawberries, and blackberries, and I may benefit by eating pomegranates and green tea. So it's not, and I hate to tell you this, human beings are the worst subjects to do a clinical study on. They're unreliable, especially with diet and food and exercise. They're unreliable, they lie almost as much as AI. So a lot of these studies are not easy to interpret. But it's a very good question. But we should be doing more. All right, work on the case. I'd love to hear your response. Okay, can I hear some interesting, creative thoughts on these questions? Anybody from this table, what do you want to say? Right, bring the mic over.
Do you want to pass around the microphone?
I want to get some.
Yeah, we can.
All right, say something.
Okay, it's kind of unfair that we have to start because we don't know what we're supposed to say, but this was very interesting, and what we decided was technology is good. Technology is what gives humans the advantage over other species, and who's there, who's going to be around to monitor this technology, and finally, if we could get a dog's personality the same, we'd clone the dog.
Do you want to say something? Oh, speaking about clone dogs.
Speaking of the dog.
Oh, what a beautiful dog. Whose dog is that?
I don't know, just came in to join us.
Are you cloned?
Charles, would you like to?
I wish I could have cloned my collie when I was a kid.
So what do you have to say?
Oh, I think technology is good, and using it ethically is good.
Do you think we have a moral compass?
I think we do. I think AI does not, so I prefer to have us doing it.
Oh, very good. Anybody from this table?
I sort of brought them down by saying that I think there are so many of these technological challenges that are probably overwhelming, that probably people aren't up to the task of figuring out what to do with them. So I think I think, that might save us, because there might be a yuck factor, that's a little simpler, that just says, it's too complicated, I don't want to talk about it. But I think I was half kidding when I said, great, now I'm worrying about climate change, and I'm worried about AI, and I'm worried about nuclear holocaust, and now I get to worry about this. CRISPR.
The Yankees are losing.
Are they really? No. But the Mets are winning.
Oh, okay.
So, but the half serious side of that is that there's really no telling how humans are going to respond to these challenges.
Do you think we have a moral compass?
I think some of us do, but the distribution of power is uneven, which you already mentioned. And so, if you take AI as kind of an analogy, there's really hardly any, if any, regulation of AI. When there was an attempt by the previous administration to do it, it was completely undone by the next administration, and they actually went as evangelists over to Europe to persuade them that they needed to do exactly the same thing. So, there's all sorts of scenarios that can play out, and I think, as a Buddhist, it's one of these things where you just trying to predict the future is just kind of a useless exercise, because you're not there yet, and it's
well, my understanding of Buddhist philosophy is that nothing is purely good and purely evil. It's both.
Right.
You got to take the consequence of both.
It's something.
That's karma.
Yeah, you don't. It is karma. Right. It's all whatever's happening.
Right. Fascinating. All right, round to you. Who wants to say something?
I'm We had a nice discussion. I think we agreed that, well, I don't know, do we agree that for medical purposes, we have a hard time seeing a situation where one shouldn't use this technology to heal someone or to prevent a newborn, from having a terrible disease. As far as morality, I'm not sure we spoke about morality, but my own personal, jump in if you feel differently, but it's a human construct and it's flawed and it's not black and white. And so we can say we have a moral compass, but our morality might be, we're a society, we have certain traits that are similar, but they're different to a different society. They might have a different set of different morality, as it were. So whether we have it or not, and also people have morality and don't always abide by it. And so, we're flawed, and, this is powerful stuff, and to put it in the hands of flawed organisms has inherent dangers.
I agree.
So, I don't know, anybody else?
That's good.
No?
You're next. Do you want to say something?
I think our discussion had two components. One was very pessimistic, and the sky is falling, and it will fall no matter what. And that nobody can be trusted, governments can't be trusted, physicians can't be trusted, researchers can't be trusted, et cetera. And then there's the other side that came up in the same conversation, is that we've seen throughout research, especially here at the laboratory and other places, that there is an ethical component and a lot of folks weighing in on what's going on, and maybe will curtail evil or negative to a point that it'll be in the same awkward balance that it is today. So,
last.
So the idea of having a baby without gametes was interesting, and so it was brought up that a same-sex couple could potentially have their own baby with both chromosomes, and that's actually kind of cool, right? That would be lovely. The idea of stealing somebody's, if somebody's, an astronaut and you just really admire them and you take some hair and you create your own baby from that, it seems like getting consent would be hard, and then you. But
you get a haircut. Do you sweep up the hair from the barbershop? No.
No, I know, but somebody, I mean.
So anybody can walk in and.
Yeah, I mean, I don't know. I'm just saying that potentially that could happen, and I think that the inequity or with access to care is a huge thing, because even now people with infertility, some people can afford IVF and some people just can't. And I worry, I see venture capitalists moving into medicine more and more, and I really strongly think that's a problem. And I can see this being something that people are buying stocks in. And
let me tell you something. What I see happening in New York, there's no more private practice. All of the private practice being bought up by these hospitals, and all of a sudden, doctors are not seeing patients for more than 15, 20 minutes. It's a matter.
Double what most of our doctors here often have.
It's
10 minutes.
It's terrible. I mean, I'll tell you.
Kira was 10 minutes.
I'll tell you, it's, in medicine, there's a principle that a parent can't be a physician for their child, right? But that's not an easy ethical issue, because who knows more about the disease than the parent, who's a physician? So I'll give you a case. I had a Columbia student. She was born with a really defective GI linkage to the from the stomach to the GI. She could not take any fluids. She had to have an IV all her life. She could take some solids, and it was miserable. She had to undergo operation, nothing worked. When she was a sophomore at Columbia, she started working with this Cornell GI woman, and they, who did these unusual operations. And she first went to her, says, "Can you help me?" She said, "No, I can't. I read your reports, I can't help you." And she worked for her for 2 years. At the end of 2 years, the student saying to her, "Now, you know me. You know my sickness. Review the charts. Can you do something?" And she reviewed the chart, says, "Yeah, I'm willing to try something." She did the operation. When I met the student about a month ago, it was the first time she had coffee at Starbucks. It worked. And she realized that by knowing the student not just as a 15, 20-minute, hour thing, but by really seeing over the course of years, she could make a medical decision she couldn't make. So to see what's going on in medicine when you go into the hospital, it's frightening. It's frightening. Anyways, this has been so much fun. I hope you've enjoyed this as much as I had, but it's really, and if my email is up there, if you have any questions, be free to email me. And especially, as I was telling Kurt this afternoon, I'm very interested in conflict. In my course at Columbia, I lie to everybody in the course. I tell them in the beginning, I'm going to lie to you to stimulate discussion. But the last lecture, I will tell you the truth. So you can ask me any question, and I'll tell you my honest opinion. But it has to be a challenging question. And every year, they always ask me the same question. They say, "Do I ever find a conflict between science and religion?" And I said, "Give me an example, and I'll tell you what I think." And I find that such an interesting question, because it really goes to the whole nature of humanity. It doesn't matter what religion, but it's really, we've been intelligent for millions of years, billions of years, and we've had to deal with this. And how we deal with these things are so fascinating to me. So if you ever come across a question in science and religion that you want to ask me, I'd love to think about it. I'll give you, can I give you one example? So there's a Jewish principle. You cannot ask God for miracles willy-nilly. So if you see an ambulance coming down your block, you can't say, "Oh, I pray to God that wasn't coming from my house." That's useless. Either it was or it was not. That's really a violation of a basic law of Judaism. Then the Talmud says, what about praying for the gender of a baby? So the Talmud says, yes, you are allowed to pray for the gender of a baby for the first 40 days of pregnancy. Now, if I would ask you, when is gender determined?
40 days.
When?
40 days earlier.
At conception. 15. Right, X or Y.
The hormones have to kick off
No.
at certain times.
Right. So it turns out we discovered a gene called the SRY gene in the X chromosome. So the default of every fertilized egg is female. And it's 40 days. This gene gets turned on or gets turned off. If it's turned on, it becomes a male. If it doesn't get turned on, this XY fetus becomes a female. Jamie Lee Curtis is a prime example. She's completely woman, and all her cells are XY. So here you have a conflict, and there is a scientific explanation. So I'm always fascinated by these things. All right, thank you.
Thank you.
Actually, Martha got me sad, was to remind us about Alam and that